he just submitted his thesis 2 days back, Shukriyo, so he's now Dr. Shukriyo Shatra, and Omurto helped us a lot with the last part of the work. And obviously, none of this would have been done if Jyotishman did not bug me and continuously tell me that I can do this. You have to keep trying, and finally, we have some results. Funding from various sources and the rest of the group, and thank you for listening.
Thank you, Devatri. The talk definitely is going to be quite quick. So let's ask the audience for other questions. Yes.
So when about that, it's like always defining the excited state in gas speed. The excited state space.
Excited state space.
Yes.
So we have the so CASPD2 basically you can target states, right? So we target a few of the low-lying states, and there we see that already the triplet pair state is there, the PU state is there, and so on. As in, you can even localize the orbitals and see what kind of orbitals are important for each of those states, and therefore you can find a diabatic picture of what that state is, that adiabatic state is made up of.
So, this means you made the ground state that you should define the gas state?
No, excited state. Excited state. So, state averaged excited states. CASPD2 you can do excited state pretty easily.
Interesting.
Yeah.
And the other question is, like you are defining the coupling in the frozen density approximation.
I am, yes.
So does those take care of non-adiabatic?
It doesn't, but I am to begin with, I'm calculating these couplings at various points that takes care of some of the, at least the geometric effects first. And then when we have actually done even some surface hopping dynamics, there we have taken care of the non-adiabatic couplings and seen that the dynamics happens to some extent. But you see, these are very big systems. To do a real non-adiabatic dynamics in a molecular system is kind of a no-go. So.
Yes, first.
So I have one question that we have. So yeah, can you just age in the age correlation part talk about the
Trimer. I don't have trimer. Yes, this is a question that arises many a times, and yeah, given that, see, whenever we have nearest neighbor and the next nearest neighbor, other than the charge transfer, everything is completely dying out. I don't expect there to be very large three-body effects. But in the charge transfer, yes, there could be larger effects, and that's the limitation of the model. We can make it better, but the moment we make it better, your calculation becomes more expensive.
The first slides you talked about the practice to the state. Where so you talked about the DP4A.
Sure.
So first you have to make the DP4A practice.
Yeah, this is a catch which I didn't talk about. In DMRG, we don't have to make the full d to the power n. What you really do is you start from, let's say, left or right. You create a subsystem and an environment. Why you need the environment? So that your node pattern remains correct. Then on that subsystem, which is done very well, and the environment, which is done crappily, you take the wave function. That wave function, you will get the density matrix. You can trace over the environment degrees of freedom. Then you will have the reduced density matrix only on the system. And from that system, you will retain the most important M states. Okay, but on the other hand, if you really did the SVD and went, if you could afford to, which you cannot, at least for bigger systems, you will arrive at the same point.
Yes, Shomla, then sir, it was a nice talk. So I have a question about the hydraulic coupling in the carotenoid system that you mentioned. So for the, so you mentioned that the high frequency modes are very important for the monomer in the initial phase.
Yes.
And low frequencies are important for the timer in the later time, right? So how do we understand this? So in the initial time point, those high frequencies are thinking more for the single frequency gap, but at the later time point, yeah.
No. So the first part, it is basically BU to AG mode. And when it is going from BU to AG, those are basically those bond length alternations. And if you think about these bond lengths, when the bond lengths change, the energetics change very sharply. Okay, so that's why these are going to be high frequency modes. On the other hand, later on, what is there? It is the AG, which is like a triplet pair on the monomer. From the monomer, this one of the triplet is going to hop to the other molecule. So in order to involve the other molecule, you basically need these kind of intermonomer breathing kind of modes, which are anyways very soft modes. So that's
I see. Thank you. Dwayne and then Gauri.
So, what temperature do you use these calculations?
0.
0. I was just thinking if you went to higher temperature, these spatial exotonic effects would vanish.
Yeah, but I don't know for the dynamics, maybe at some point we will incorporate to some temperatures, but as of now, we are just looking at the spectrum of energy. So Hamiltonian diagonalize.
Okay, so you can't like do it
No,
Boltzmann occupation, include those ground states and.
we haven't, but
Please, if I can, magnetic field effects. Can you include magnetic field effects? Because you can tune the triplet energy.
yes.
Because that would let you disentangle the vibronic from spin on the couple.
That's a good point. Yes, maybe yes, we can.
Okay, that would be interesting.
Yes.
Because there's a huge debate now on quantum birds.
I know. Yeah, there was the cryptochrome things.
Yeah.
Actually, so each of these, so this is what I want to say. Each of these Hamiltonians we are building, they're kind of tailored to a problem or the other. Because the full these are effective Hamiltonians. So essentially, we have gotten rid of too many degrees of freedom, and retained only the most important degrees of freedom for that problem. So once we go to some other problem, let's say a magnetic, we will have to redo the Hamiltonian.
My question is about the aggregates. So already the molecules are interacting with each other, and we have new states are formed, right, in the aggregates. Now we have to compare the ground side of the monomer states or the aggregate.
I didn't understand the question.
So the aggregated systems.
Mhm.
So now, anyway, the aggregates have different electronic states, right? So we have the aggregate.
Uh-huh.
So now, the molecules are interacting.
Mhm.
If we compare some monomeric units or the ensemble of aggregates where you have multiple.
Yeah, so basically, do you want to go back to the local basis or not? Even if you have an aggregate, do we really care about what the molecule is?
Without the local basis, I cannot form the Hamiltonian. That is just a basic problem I have. Without the monomer, I cannot form the Hamiltonian. But once I have formed the Hamiltonian, once I diagonalize it, I will see that the states that are formed are not on the monomer. They are like spread out. In everywhere. Whichever state that you have, they are spread out.
We have time. We have so much now we're thinking about the triplet.
Yes.
In a given aggregate, the different molecules.
Yes. Triplet need not be just triplet here, triplet there. It could be like 0.3 of triplet here and 0.2 of triplet here and point whatever of triplet there. And it could be it's spread out. That could happen, and that does happen.
Then when compared to other pumping process, like other productive events of the single molecules.
Okay.
So now all of these are repeated.
If you incorporate those states, for example, let's say a singlet to a triplet, which is of course spin formula, but or singlet is another singlet. If I incorporate those in my Hamiltonian, they will occur.
Oh.
There's nothing stopping it from occurring.
But in neither calculation, they are not.
As of now, because I was using ACs don't have only one singlet that is important. Because I actually started with a system that is simpler, so that I have a handle on what is going on. Carotenoids are a bit harder.
Yeah.
The next thing would be to put it in carotenoids.
Yes, Steve. Yes, sir. I think it's a similar question, but you said that you can forget about the energy criteria.
Yes,
Exactly. I'm a bit worried that then people will interpret that as actually you don't have to conserve energy. No. In this field.
I'm just saying, yeah, I know where this energy criteria came from, but a monomer, when I initially excite a molecule, it's not that only one monomer excites and the rest of it is like dead. That's not true. And when a triplet pair forms, it's not like the triplet only forms on two monomers next to each other. They can be spread out or something or the other.
When you have that case,
Yes,
that also sort of manifests in the electronic spectrum of the.
it should.
in the singlet.
It will.
So,
Yes.
so we know when that case is.
Yes, in fact, when the coupling becomes stronger, essentially, if you bring the molecules closer and closer to each other, you will see that the Davidoff splitting will grow. And at one point, you know that there's going to be funny stuff happening. But here is the catch. See, you are looking at only those. See, when does singlet fission happen? When there is coupling. And on the other hand, you want to take the monomer energetics as your energetics. That's kind of
The problem.
that's the problem.
Uncle?
Yeah, so for the coupling system, was there an experiment which was done with like the thing that when they were not linked?
No, all linked. All linked, different
Yeah,
linkers.
because I'm wondering, the fact that you were saying that it's more difficult to handle, maybe that the linker, like identity or the, or it's like psychoactivity kind of thing, right? It might have some effect on the.
No, the calculations we did were without linker. We were
Without-
just with the distances, as if the linkers are just modulating the distances. And we still found that the time scale patterns, those we could replicate. So it was driven by mostly distance. I cannot say 100% because how can I? But mostly driven by distance or average, because there were some which looked like this orientation, those kind of. But because they are much closer than you would have if they are just two monomers, so then we could actually see the manifestation that these energy rules are breaking down. Otherwise, what happens is the energy rules is kind of okay to whatever accuracy we have. So we kind of say, okay, that's fine.
So nice question. So when you're doing the calculations, can you constrain your distance then to match the distance of you are doing that,
Yes,
right? You're
we are
constraining
doing
it.
Yes, they gave us the distances and we put the distances.
Okay. I will just ask one question. Is it possible now to, with Omoto, if you guys have figured out this, that you can look at the collapse dynamics of the TT pair state under the consideration of a large spin-orbit coupling? So one of the things would be that if the TT pair collapses, it can form a single T and an S0. And this could be an intersystem crossing triplet,
Not
which
yet.
is not.
We are just looking at short time scales where the triplet pair is forming. That's the first goal. Once that happens, we will incorporate the rest.
Okay. Fine. Thank you, Devashni. That was fantastic. So could you please come have a picture? So maybe
I stand
I think you can please. So so this is the art collection book. It's something that I think Devashni has not seen. So
Thank you. I have my pen carried.
okay, so I think we move on to our last speaker in this session, Professor Devrata Maiti. And yes, maybe you have the pen drive. Which one? This one. So now I take this off. Okay. Yes.
point. Okay, so again, a two-line introduction. I don't need much introduction for these folks. Devbrata might be, it's very hard for me to say Devbrata, it's Devu, actually. So Devu has been one of the exponents of developing organic reactions, and especially CH activation reactions for a long time now, almost like 14 years he has been in India, and he has been doing this with a training that composed of very interesting bioinorganic chemistry or organometallic chemistry with Ken Carlin, and then or pure organometallic catalysis with Steve Buchwald in MIT. But he came with this fantastic idea that he is going to use palladium chemistry to do remote CH activation, things which sort of look very difficult on the paper, but you sort of have to imagine how the catalysis world has to think to do an activation reaction which is very far away from a sort of functional part of the molecule. So typically, wherever the functional part or the functionalized part of the molecule, people used to attack that site and do an activation close by. But they said, well, can I do remote activation using the same kind of a functional site? So this is something that I heard from him in a meeting that was organized by IIT Bombay in Kaleidoscope for the first time. And this is the time when I recognized that Indian counterparts, unlike some of you guys, don't get the benefit of the doubt that we do good science when we try to publish in top journals. And he had this result in 2014, where he had done a para-CH activation for the first time. And while the reviewing process was going on, there were difficulties because the leader, I'm not going to name the person here, was trying to block the paper. And this is something that ultimately was published, and that started an entire new idea of how to do remote CH activation. So there was a tremendous imprint on the organic chemistry community in that way, and we are extremely proud that we have him here in India. So therefore, without further ado,
Thank you, JD. I think this is thank you, everyone. It's really an honor and pleasure to be here, JD. You have been one of the closest friends and one of our cheerleaders in the Indian scientific community. Your presence and encouragement has always, I would say, enlightened us, invigorated us. Today, although I will not be talking about the CH activation the way you have described, I will try to take a dig at something little different that we have been doing for some time with very little success, and maybe we are trying to see where we go from here. Okay, let's see what we can talk today. Today, mainly, I'll talk at the first part. We are doing what we are doing on artificial metal, especially in here at TIFR. As you know, you have tremendous activity in this domain as well as some of the biological studies that both in chemistry and biological sciences we are doing. So what we started thinking, of course, without any formal training in this area, that can we try to do synthetic chemistry inside the engine? Of course, I'm sure you will hear later on from our very own Abhisigda, how to do these reactions efficiently, how to correlate with engine. But for a synthetic chemist like us, we thought that synthetic systems are really lacking in terms of efficiency. If you look at the engine can do one reaction very efficiently. You look at the turnover number. Turnover number are 28 whatever digits, right? So, or for synthetic chemistry, one cycle. It's great. Sometimes part of that when I was doing let's say PhD, I was getting half a cycle, not even full one cycle. Truly, that's what kind of pushed me out of that domain, that I want to have little bit more than one, right? Maybe 100, 1,000, maximum 1,000, maybe 100. This is where I got into, let's say, more of a organometallic catalysis. But still, we are struggling, as I mentioned,
half a catalytic cycle to 100 or maximum 1,000. The best of the best catalytic cycle in synthetic domain are still limited within 1,000, the turnover number, so to speak. So the catalysts are not really effective. There are multiple reasons. A lot of these are related to the so-called catalyst poisoning or the dead intermediate or dead side product formation. Sometimes the dimerization, trimerization, oligomerization, precipitating out, you name it. There are multiple problems that can happen for a small molecule when it is doing the catalysis. On the other hand, in the enzyme, as I said, one reaction, efficiency is unmatched. You can blame it to the nature. Nature has done it since time immemorial and mastered how to do it, right? But synthetic chemists like me and then some of us, I believe, we are very, how do I put it? We are very disturbed and we are very demanding and we want every reaction, right? Every reaction, every type of reaction, but nature does few reactions with efficient transformations. So this is where we started thinking, can we somehow do these reactions, some of these reactions inside the enzyme? Not only doing these reactions, a lot of these reactions, as you have seen and will be seeing again, that these reactions or the products essentially either left-handed or right-handed. So there is chirality. Can we do asymmetric catalysis inside the protein? Okay. Of course, nature does it again very beautifully. For many of the metalloenzymes or enzymes, so to speak, they can do this left-handed compound versus right-handed compound, not both of them, only one of them, one product, mind you, only one product. Nature can do really brilliantly, as I was trying to impress upon you. Now, as a synthetic chemist, I want every reaction to be inside the enzyme. And not only one product, we want hundreds, thousands of products, because those products are going to be utilized in, let's say, drug discovery program. Can we get millions or hundreds, thousands of products at least, which can then be introduced in the drug discovery? Okay, as a synthetic chemist, that's what we want. Okay. Now, if you look at the way enzyme does this chemistry, you have a metal center. If you are dealing with a metal enzyme, you have a metal center. You have already seen these metal centers and the porphyrin or heme system. I'm sure you will be seeing again, hopefully. So this ligand system is achiral. There is no left-handed or right-handedness into these ligands. The metal itself is, of course, achiral. This entire system is achiral, but the product coming out is chiral from these active site, because the second coordination sphere is chiral. That is built up of amino acids, peptides instead, like protein residues. So metal ligand complex itself is achiral. Let's say I am in the metal ligand complex. The room, this is chiral. The room is either left-handed or right-handed. Now, of course, the way synthetic chemists like us, we try to do is, we make myself chiral. The metal is achiral, but the ligand that is holding the metal is chiral. And that's the way synthetic chemists are doing the chemistry for decades now. That has certain limitations. The way synthetic chemists make the catalyst chiral, like myself chiral, that has its limitations. Some of the things we cannot do efficiently. Some of the things definitely we can do. But this is where we started thinking, of course, this field is evolving at this point, where can we do the catalysis inside the room, where catalyst is there, but the chirality of the room will be transferred into the product that is going to be formed. If I, for a moment, imagine that each of us over here are one product, can we get selective product formation efficient? Of course, turnover number, frequency should be high, as well as each of us has to be a chiral molecule. Can we produce out of this room? So this is what we started thinking about. What we thought that we will not get into the protein which has already metal in there. We'll take a protein which is, of course, chiral. The pocket is chiral. Can we bring a metal center inside this, which
we have some control over. This is what, let's say, JD was saying, that we have been designing ligand metal complexes. So we know how to control the properties of the metal. Okay, but can we bring it inside this room and start doing the chemistry? So we wanted to essentially merge the base of both genetic one as well as the synthetic one. Synthetic part, we have had some experience training, so we know how to really control the properties around the ligand metal complexes. So we needed to learn the biological part. Of course, we started learning it. This is the same cartoon saying that inside the protein, we would need an anchor to bring the metal complex. Okay, this is, of course, not an absolute new concept. It has been known, and few leaders like, let's say, Francis Arnold group at Caltech, for more specifically for this domain, Professor Tom Watt group at University of Basel, been doing this. We have relied on streptavidin, again, a known protein for some of us, I'm sure. It's a very steady protein. It does not, behave weirdly at a little bit higher temperature, meaning that synthetic chemists like us, we can deal with this protein. It's not going to create much problem, right? So inside this protein, essentially, we decided to bring in this organic molecule. This is a tetrameric protein, and we are going to bring this organic molecule. Now, this organic molecule has an organic part, the biotin part, and a metal part. So you can say that it's a metal profile. So this is the ligand. Again, the ligand designing we can do, we have been trained to do. And of course, which metal, what chemistry, we know what to do. And what type of organic molecule, like each of us, product we want to get out of it. Therefore, the design or the choice of the metal and the immediate ligand is in our control. We have some exposure as well as literature knowledge of it. We introduced this biotin part so that inside the protein or inside the active site, this will be anchored. Of course, biotin streptavidin interaction is legendary. Right? This is almost like a covalent bond. They are not covalently linked, but this hydrophobic interaction and hydrogen bonding interaction and many other weak interactions put together, this is really one of the strongest non-covalent interactions you can find anywhere in the literature. Therefore, once again, you take the biotin, you figure out a way to attach your ligand metal complex. Once you are able to do that, we know what type of metal chemistry happens here. Since now this metal is not in solution, but inside the protein, so we can use the goodness of the, battery part. Okay. We can use the goodness of the metal chemistry inside the protein, and we can expect the asymmetric catalysis to be happening out of this metal center. One good thing happened, or when we were trying for long, we realized that we can crystallize this protein. Again, this is to some extent new for all of us. Of course, we took help from our colleague, Professor Rosin Homing from Bioscience Bioengineering. And this protein structure essentially clearly shows that the biotin and streptavidin, their interaction, and clearly demonstrate where our metal ligand complex is. This, of course, without getting a crystal structure, we can also do the simulation docking studies to find it out, but getting a crystal is nothing like it. You start believing it, right? More than your computation. Of course, I believe my collaborators and computational, but having a crystal structure, nothing like it, right? This is where many of the synthetic chemists' eyes get lit up, right? So this is what we got. One thing we realized or noticed that between this biotin and this metal center, this is long linker. That means that metal is not necessarily over here. Metal is also flip-flopping over there. So it's up and down, up and down. It's able to flip-flop. Sometimes metal is down, sometimes metal is up. But more importantly, by seeing the crystal structure, we immediately recognize that this 49 position asparagine, 112 position serine, these two are going to be. Sorry. Yeah. So these 49 and 112 position seems to be the one where we would have the maximum effect from these amino acids for the chemistry that is going to happen at the moment, right? Of course, there are many other amino acid residues. We need to have some sort of hypothesis as well. So this is where we ended up having that these amino acid residues we want to change. In addition to that, we realize this 121, that is this
It's a printed here. This device
The wall.
on the wall.
just
Yes, got it, got it. Actually, this is my favorite board. Okay, so these two amino acid residues, we knew that it is going to affect the catalysis or the chemistry happening to the center. But this residue, we wanted to modify site selective mutation. We want to change one amino acid residue at a time, two residues at a time, three residues at a time. We have not. And we initially over here, we were trying to learn these mutations with Samula Majumdar. So eventually we learned it. So how to change these residues one at a time, two at a time, three at a time, not only these, maybe also the other residues. Okay. To keep the long story short, when we were able to do that, we learned that these reactions, so this is an organic reaction, it involves the carbon-hydrogen bond activation from this stage, but we were able to show that this product is forming. Of course, this product forms without protein. You don't need protein to form these products, but more importantly, we find that when we do not have essentially any modification, these reactions was with very little energy flattening, meaning that 50-50 left-hand and right-hand compounds are formed if you do not change the protein. If you do not do any mutation, it's a 50-50 mixture. If you do not have the protein, of course the reaction works, but once again, it is going to be 50-50 mix. As I was mentioning, those amino acid residues were critical. We started systematically modifying. Of course, with some hypotheses we have had, but what we realized, and of course, a little bit at least we hypothesized, that we need to locate the metal center because metal center was above the plane as well as below the plane. This is a cyclopentadiene CP star essentially. So with respect to this ligand, the rhodium, as I was telling, would be above the plane as well as below the plane. Now we figure out if we introduce the pi stacking interaction, therefore introducing this replacing serine with the tyrosine at this 1-1-2 position, and if we are promoting the pi stacking interaction, we could place the rhodium above the plane firmly enough so that no longer we are getting both the left-handed and right-handed product. We get, let's say, near exclusive or as exclusive a synthetic chemist or an enzyme specialist can get. Almost exclusively, we are getting one product, right? So the modification is, I would say, almost two modifications. This is required for the carbon-hydrogen bond activation process, mechanistic purpose, the glutamate. Previously, we were having glycine, lysine, now we have glutamate. And in this position, from serine to tyrosine. That's all. This double mutation made sure that instead of 50-50 mixture, we are getting only one product, right? We started doing a little bit more and started thinking, how can we really extrapolate? Of course, once we are getting one product, since it's a man-made catalyst, not only one example can be shown. As a synthetic chemist, we can then do 50, 100 different molecules if we want. Okay. Of course, these sort of motifagi will come in a moment, are prevalent in many drug molecules, either which are approved or under a clinical trial. So this is one thing to get R-selective or one-handed product with this catalyst by double mutation. These two sites are mutated. But we thought that can we not get the S-selective product? In synthetic chemistry, what we do, if again, I am a catalyst, I have a metal and ligand within me. If the ligand is left-handed, let's say the product is left-handed. If ligand is right-handed, the product is expected to be right-handed. This phenomenon is called stereodivergence. So you control the ligand chirality, you control the product chirality. Now, as I told you, for the protein, the whole room is chiral. And this is made of amino acid residue. If I have to change the chirality of the room, I have to go back and fix the chirality of every amino acid residue. But that's not going to happen. And definitely, all the budget of Indian government funding agency will run out to change all the amino acid residue and reconstruct the protein with the unnatural amino acids. So what, at this point, we realize that since the efficiency or the selectivity for the R product is due to this pi-stacking interaction, having a tyrosine here and the CP star unit, and that's essentially immobilizing it. Can we not just flip it? So can we get the S-selected product by putting a phenylalanine at top? As I said, that these two units are very close to the active site or the metal center. So this is what essentially we did. We went back to the native or the serine. We didn't do anything at 112. We kept as it is. We did just a mutation at the 49 position. And this is sufficient. Now we find to give the product at this center for this point chirality to be excellent. This was something then set up for the stereo divergence inside the enzymatic system, which is not absolutely unknown, but I would say rather rare. And controlling such stereo divergence or producing product in such situations are relatively rare. Okay, of course, this is just to show that when you take wild type without any change of the protein residue, you get 50-50 mixture. This is the HPLC trace. When you have this double mutation, as we were talking about, you get R product exclusively. We have the crystal structure to back up. And when you do other double mutation, as I discussed, you get the other product. So this is a 50-50 mixture of left-handed, right-handed molecule. This is 100% right-handed. This is 100%. Okay. Of course, as you could imagine, being a synthetic chemist, if we now have some sort of control, how to modify the protein and how to take the metal complex inside the protein, we can now imagine or reimagine how to make hundreds of molecules if we have the short sets funding and resources in terms of this is what we've been doing in terms of synthetic chemistry inside the protein. Of course, as you imagine, there are many interesting molecules one could look at. We started designing and preparing and scaling up some of these molecules. And there are some interest, but really taking some of these approaches to the industry level and making, let's say, that some sort of drug discovery program with these will be exciting. I did not discuss the reaction mechanism in greater detail. I can come back to you if you have more questions. But very simply what we realized, because in this domain we have been working for some time, not in the enzyme, but outside the enzyme in the normal organic solvent. And some of these are either literature-driven mechanism or homegrown understanding that we have over the years now. So we have a metal center, and this is the ligand to start with. And we have these, the substrate, organic substrate, the CH bond at this position, in this case, not the distal, but the first position CH bond doesn't happen with the help of this carboxylic acid. This is what as I was trying to mention in the previous slides, that glutamate unit, right? So we needed this glutamate unit because the CH activation is dependent on the glutamate. So this is particularly where, let's say, palladium acetate, when we do the palladium chemistry, palladium acetate is required, not palladium chloride. Chloride cannot participate in this bimodal activity of CH activation. You need an acetate, and this is where we were doing glutamate. Glutamate otherwise does not participate in the in any other process, but critical CH activation step, you need this glutamate. Okay, so that's the third point of mutations that we were using. But once we are able to activate and then place the metal inside, of course, you don't need protein for this. You can do it anyway in the flux, but protein makes sure that this differentiation in terms of the CH bond getting activated, and specifically the product formation step where the stereoinduction is happening, enantiomer determining step where, let's say in this particular case, the second molecule, so first molecule getting activated, second molecule getting incorporated or interacting with the metal center, the nitrogen exclusion and metal carbon formation happens, and this is the stereodivergence, or sorry, this is the enantiomer determining step where you are deciding which product for left-handed or the right-handed product. Again, this is enzyme is essentially controlling it, and overall this product formation then going on. If you have more questions, of course, we try to study these elementary steps in greater detail, how exactly it is happening, both by computational studies in collaboration as well as experimental studies that goes on in my lab. Okay, so in the interest of time, I will move on. How much time I have?
We got 10 more minutes.
10 more minutes. Okay, great. I'm even more excited. So this is again, this is something, both of these areas are something I normally don't talk about, and we don't have a lot of result to share with. Well, it gets me going or thinking, interaction with my friends over here and in other places. Can we try to do something in the drug discovery program? We are synthetic chemists. We know that so far from India, not even a single drug being discovered. And typically, you need 10 to 15 years and several thousands of dollars or no, so it would be billions of investment. So thousands of crores investment for a single drug. Okay, this will be audacious of me or of anyone to really think that we will definitely do it. But without taking very step, I think this is also not fair. So we started thinking, can we not get into the AI/ML based or driven drug discovery program in our own laboratory? Of course, we needed to have some collaboration. I will come back to that in a moment. So what's first one?
Yeah, we take the.
Okay. Okay, maybe I'll just use this. Okay, so this again, the topics over here is very much popular. Many of our colleagues over here are well versed with, and they have been practicing this throughout their career. We are, so to speak, novice here. But what we thought that we will target the PDL1. PDL1 protein is overexpressed in number of cancer cells. We thought that if we can generate or if we can come up with an inhibitor for PDL1, perhaps we would have an opportunity to look at some of these drug discovery programs. Of course, PDL1 inhibitors are also well known in the literature. Approximately 300 PDL1 inhibitors are known, and their corresponding IC50 values are known. What we started doing in our laboratory is to develop AI/ML model which fits the structure of this PDL1 inhibitor and correlate it with its activity, that is IC50. Once we have the right AI/ML model, we can have the input and output correlated. Of course, out of these 300 molecules, 250 we have initially taken as a training set, and 50 we have taken as the test set. Once our algorithm is right, and being at IIT Bombay, we get, of course, the best of the best undergraduate to integrate with us in these activities. And that's what we have been doing. But we realized that, of course, we can have a generative AI model to start having compounds which are newer in the domain. Just to give you the pipeline a little bit in detail, we, being chemists, we love the chemdrop. As you would know, again, some of us are very familiar with this workflow of the pipeline. We, as chemists, we are stuck with the chemdrop. We don't know how to convert it into the machine learning form or the computer readable form. Computer can draw the chemdrop, but linking that chemdrop back into the computer so that computer can read in a multidimensional field is not really trivial for synthetic chemists like us. This is what can be done by SMILE. SMILE takes the input, whatever input is here, the bond connectivity angle and everything possible. And then we started feeding these into the large language model. Of course, you can have a different model like graphical network, GNN. You can have whatever model you prefer to have. But we started believing more on the ChemBART, which is a large language model and specified for the chemical structure. Once we have started feeding the structures of these 300 molecules, let's say, with the ChemBART model, we started, of course, getting the embedding. So the chemical structure is now represented in a higher dimensional form, which is, again, some of you are very well versed with. For me, it was a learning phase, and it will remain as a learning phase. So once the embedding is done, we can then start looking at essentially contrastive learning, which is clustering of the potent molecule compared to the less potent molecule. We have done that, and we find that, let's say, most active molecules are together, less active molecules are separated out. And once we have this contrastive learning or the clustering done, then we can essentially feed this data into the Gaussian mixer model. Once we have the Gaussian mixer model, every point of that Gaussian mixer model is a new compound. So 300 molecules input is going to give us tens of thousands of molecules as an output. Now that's too much. And again, I'm a synthetic chemist. Our group is synthetic chemist group. So we cannot essentially rely on these tens of thousands of molecules that is generated by this generative system or AIML. So we started looking at it further. Before going into that, before contrastive learning, the more active and less active molecules are all over the place. After contrastive learning, essentially you sequester them and then you do the Gaussian mixer modeling to fit it, right? That's what we have been doing. Once we are able to do it, as I said, from 300 molecules, we have now tens of thousands molecules, which is predicted by artificial intelligence machine learning model. Now, what do you do about it? These tens of thousands of molecules, naturally, I am not going to synthesize, and this defeats the purpose. Of course, we have to pass through all these molecules through the molecular selection criteria, something, again, our biology colleagues are very well versed with. We started learning, of course, passing it through the docking score and Lipinski rule of 5 and all the drug likeness property. We essentially come up with, let's say, 10 molecules. Out of 30,000 molecules, we essentially come up with 10 molecules. Let's say some of these molecules are shown in here. Again, as a synthetic chemist, we now are synthesizing these molecules. And we have a biological team from BRC as well as Tata Memorial Center, ACTREC essentially. We are working very closely to synthesize first in our laboratory and then check out the effectiveness of these molecules. Okay. So this is, again, an open-ended program we have entered into for some time now. The last few minutes, if I have, I will discuss more of a personalized medicine. Again, these are the topic. Being a synthetic chemist, at points we are very far from it, but I think we are very excited to figure out if we can contribute at least to some extent to this domain. As we all are aware about, let's say particularly for cancer treatment, for a given cancer, if a medicine is given to, let's say, 10 different patients with the same problem or the same disease, it works for 2 or 3 patients. It doesn't really work for, unfortunately, 6, 7 of the patients. So there is an absolute need to integrate the patient-specific data into the drug discovery program, so-called personalized medicine. So this is where we started thinking, can we not utilize some of the drugs database that exists over there? Can we integrate essentially transcriptomics data with the IC50 value of the compound? Again, this workflow or the pipeline will be similar as we have described earlier. So this transcriptomics data, essentially also another problem is these are very specific, or the data which are available are not India grown. We have the data, these are mostly kept either in US or Europe, but our country specific data are missing. But nonetheless, we started collaborating with Tata Memorial. They have some sort of data, but not a large database. As you know, for AIML, you need a large database to train the program first and to get the output in a more reliable way, better the data or more the data, it will be more useful. But nonetheless, we started collecting the data available in the literature or the open source database. We started converting the transcriptomics into the embedding. Okay, I'll come to that in a moment. Of course, in some of these cases, the drug molecules are known and their efficacy for these particular patients are also known. What is not done or not done in a great extent, how these drugs are correlated or their efficiency is correlated with the transcriptomics data. So this is where we essentially have two pipelines. One pipeline is training the drugs into the embedding space and bringing them in the latent space of both of these and then fusing them together. This is the two data set we are talking about. This is again open source database, TCGA and GDSC. You can have access to this. TCGA is essentially dealing with only the patient's transcriptomics without any data for the patient-specific drug efficacy. This has nothing to do with the drug. This is just the transcriptomics of the patients. GDSC, on the other hand, is a bit more useful, but the data sets are limited. For, I would say still it's a good number of data with a variety of cell lines and almost all the known anti-cancer agents. These studies are done. So GDSC data set has the transcriptomics data as well as the IC50 value for a given drug, which is already in the market. And therefore, again, I'll come back to the same place where we started. We started taking the RNA sequence and encoding it. And of course, reducing some of these parameters, we started having this latent space where we can essentially fuse the latent space of this gene expression program with this molecular encoder. And again, these two latent spaces can interact with each other, can give us a new molecule through a decoder. This whole exercise is trained as well as checked by having the input data from those atlas that is existing over there. Once we have the generated molecules, once again, either we can synthesize those, or at least through this drug likeness test, we can tell whether we should have a reward or we should have a critic model placed in it and then place overall for the further development. Okay. So what essentially we are looking for, therefore, the bigger idea or bigger inspiration for us essentially is, let's say when a patient is admitted to the hospital, if we know the transcriptomics of the patients, which is not that very difficult to know, this is one of the first few tests that is done at the hospital. Based on that transcriptomics data, can we predict which medicine will be likely to be given to that patients from the existing drug? If we can do that, that would be one way to do the treatment. Another would be if there is no existing drug that is going to be effective, can we not then suggest something likely to work outside the, let's say, approved molecule, and can it be then expedited? And this is where our India-based data set, which is essentially the phytochemicals, can we take into the phytochemicals? Phytochemicals are known to have less oxide toxicity. Perhaps that can also be integrated with this overall pipeline. So again, there are multiple interests in this domain, multiple collaborations are required, and this is where we are driving forward. And we also would like to have essentially AI-based identification of the phytochemicals for the treatment. And this is not necessarily has to be related or limited to the anti-cancer therapy. It could be perhaps extended in many other ways. Again, for me, these are all over last several years now. These are a new learning curve, and I'm pretty excited, mainly because none of these are done by me. My collaborations as well as my students. The more specific collaboration is with Professor Dr. Vikram Gota from ACTREC, which I realize is a sister institute of the right. Dr. Vikram Gota and his team essentially helping us out with the biological studies as well as they are ready to do patient level study if anything comes out to be effective. And of course, in our institute, we are also collaborating with Dr. Shivaram, and we are also interacting with some of the giants of the AI/ML drug discovery as well as the medchem program. And we also have collaboration with the BRC scientists, particularly Ovik Chaudhary. Anirban is one of the main driver in the laboratory for doing this. Of course, we have Somnath, which is again who are really setting this collaboration between IIT Bombay and BRC, and he has every reason to come to every day in my lab because his wife works in our department. So that works out quite well for us at least. And then, as I was telling, the main players are the undergraduate of IIT Bombay. Apparently, they know what they are doing. We will rely definitely heavily on that. With that, thank you, JD. Thank you all for listening to me.
Well, wonderful, Devu. I think lots of questions. Yes, we'll start with 14.
I really exciting stuff, especially the first part actually. So if I understand correctly, you have your cluster based on which mutation you're using, it's oriented radiotherapy, right? But it looks like you mentioned that glutamic acid is only 121. If I'm not wrong.
Right.
So how is that just, I think it seems slightly far from the cluster.
Right, of course.
How is that actually doing actually both the stereotype because it's really far from the cluster.
Right. This is true, but I didn't really discuss. This is not a monomeric protein. This is a tetrameric protein. Essentially, it's, for the simplicity sake, I kind of implied that this is from the same monomeric unit. Since this 121 from the second unit, from the neighbor that comes in. Of course, I didn't discuss.
Okay, you're saying basically the binding site etc. somehow relatively.
Right. This we also have the protein structure as I was saying, which is clearly it's not the 121 glutamate is not from the same unit. It's the neighbor that coming and pass.
Yes. You have someone from the neighbor.
Not from there. But it, once we try to draw the neighbor, it becomes so complex. It doesn't look that we can present.
Yes. There's a student question for us. Ayush.
So, first of all, nice talk, sir. So, my question is that how we are experimentally ensuring that substrate you are choosing is going to the active side of this artificial movement.
So how are we making sure that the substrate means the organic
The
compound?
organic molecules.
Yeah. So again, it has to be done. Well, first of all, the protein that we have taken, it has the cavity size and everything is big enough. This is where the streptavidin essentially plays into our hands. This is not a very specific, protein. It has binding pockets which is like very big enough. Has it been any other proteins? And the channel to reach to the active side is really congested, then we would not have been able to. So the chemistry part is trivial, let's say, to start with. The chemist, once the substrate reaches, chemistry will happen. But your question is how the substrate reaches up to the
non-polarization that gets.
So this is where we didn't really take a very, let's say, rather complex or very, very involved protein here. Here, the protein itself, the streptavidin, is pretty much accommodative to many of the organic molecules. And this is kind of, I would say, in many different domains, it is already known. Right? So it's not of a selectivity issue based on the substrate binding pocket. The substrate binding pocket is large enough, it can bind enough substrate. Right?
So, and what is the unknown number you are achieving?
Oh, yeah. Well, I didn't show over here. It's tens of thousands in some, depending on the substrate, of course. So we do have tens of thousands, not in the order what enzymes are going to do, but not definitely not 1 or 100, right? It's much higher. Okay, 10^2, 10^3.
Okay, I'll come to Tosha. I think there are questions from Ankuna and Devashri and Deepa. Devashri.
Okay, one question is kind of related to what was asked, how the substrate can bind into several substrate can bind into one pocket. My question arises from, and maybe it's just that, if you have a more tighter binding pocket, essentially your chemistry is more directed, catalysis, blah, blah. On the other hand, if you have more floppy, then more things can come and bind. So
That's right.
how do you solve this catch-22 situation?
Okay, as all of us know that natural enzyme does one thing very efficiently, and therefore everything is really picture perfect, right? Absolutely perfect. Over here, we are not really looking for that. We are not looking for, let's say, 10 to the power 5-fold turnover number or 6. We are happy with 10 to the power 3, 4, not 6, 7, 8. We don't have to reach there. So therefore, the substrate promiscuity or the diversification is something we would like to achieve. And we are okay to compromise the turnover number or efficiency to some extent. So this is definitely compromised version. If we end up promoting more mutation, this will be possible, but then the substrate scope will miss out. Being a synthetic chemist, I value also my substrate scope in addition to the catalytic reactions. So this would be somewhere the compromise state, where substrate scope is large, efficiency is all right, not competing with the enzyme-like activity, but much better than the synthetic. That's okay. Thank you.
Yeah, post extending with the RNS isomers, you are the same turnover number. I was
no,
wondering with the
no.
dynamics.
No, this is dynamic. It's not going to be the same, right?
Right.
It is the same. Even that we struggle actually for the S-selective product. Over here, we have very high enantiomer selectivity. Even the enantiomer selectivity and turnover number relatively better. Over here, we have relatively less selectivity. Let's say enantiomer selectivity 99 to 1 over here, the best case. Over here, it is more of a 90 to 10. So the enantiomer selectivity is not that great. Let's say if it is 5,000 turnover number, we got maybe 3,000 or so turnover number in the best case scenario. So it's not absolutely hand in hand, or the inversion or stereo divergence is okay, but the efficiency is never the same. But that also tells that, see, only we are doing 3 mutation at best. If we want to invest more, and if anyone is showing interest in any of these products, perhaps the best would be to go and do a little bit more mutation, just not 3 mutations, right? For a synthetic chemist, again, I'm little bit, my group is, they are having fear of these mutations way too much. They don't want to do. They want to see their reactions. So there are different mindsets. If someone over here who are very comfortable in creating, let's say, more of a random mutation, this is what it would be, right? In a lab like, let's say, Francis Arnold or so, they have directed evolution. They have access to so much thing, and it's expensive. This is done almost, let's say, without any budget. So you have to carry on, or you have to weigh on different options here. But the best would be, coming back to the first question also, the best would be keep on doing mutation to make it perfect. But we not necessarily want to go absolutely over there. We want to have a compromise situation and get the best of it.
Because in heterogeneous metabolism, you have to find this sweet spot. Then you have lots of ways of measuring density of, just the intermediate binding energy, doing a working of all of this. So I was wondering if that was something that could be approached with this sort of homogeneous setup.
It can be, but I would say we are not gearing up towards that, at least at the moment. But hopefully in future, if you are excited about it, we can definitely talk more on that. Yeah, thank you.
Okay, so questions, Roshan first.
Nice answer. I just wanted to know that how do you decide upon the amino acid? Like aspirin will give R and phenylalanine will give S.
No, we didn't decide whether it would be left-handed and right-handed product. It's just a pure observation that this is what has given. But then once you have the observation, you can retrofit why it is happening, and which we have done. But we were not in a position, or we didn't think ahead that whether this should give R or S. But once we have the result, then you can build up the model. I mean, this is not always the best way to do. It's better to have the prediction first. But in this domain where product formation is the critical step, we did not go for, let's say, prediction prior to doing that.
Okay, Steve, and then I'll go ahead to you.
Also on this slide, thank you, very fascinating topic. Is there, you've got a very large switch in North America, but the structure change does not seem so large between the two. So do you know why? Is there a relationship between the orientation of that sample?
So I tried to briefly mention that it's essentially this tyrosine group, the pi electron cloud of this tyrosine ring and this CP star unit, they are interacting. This interaction mean that the rhodium can be only on top, rhodium cannot be. So this pi stacking is important. When we have taken this unit and put it over here, essentially not the tyrosine but the phenylalanine, then the phenyl ring and this pi ring are interacting over here. So this one we have removed now, of course, by mutation. This interaction was sufficient. Now this unit is essentially shifted over here. That shifting was sufficient to make sure that this pi stacking is happening. Some sort of interaction, it need not be very strong interaction. Some sort of pi interaction is happening and therefore rhodium is placed below. And the good thing is, of course, we do have the crystal structure also for this, right? So we have the wild type crystal, doubly mutated in this form as well as doubly mutated in that form, which clearly shows that in the wild type, when nothing is there and nothing is there, without any of these mutations, we see that rhodium is up and down. Some of the, in the protein structure, you can figure it out. But once you have these mutations in place, you see that rhodium is fixed. My orientation, let's say if I'm the catalyst rhodium, my orientation is this versus my orientation is that. So it's one orientation. That's what is driving the inertial activity.
Is there a sort of a theoretical relationship between that orientation and the, I suppose, the surrounding?
Yeah, I mean, yeah. So, this is again the synthetic part. If you essentially do the catalysis from top phase versus the bottom phase, if you are getting the same compound, I think you can get the
access